Hey guys, welcome to my channel where I explore cutting-edge scientific advancements and their impact on society. And in today's video, I'll delve into a highly controversial topic, which is CRISPR babies. And I'll tell you everything you need to know regarding the concept of genetically editing human embryos using the CRISPR-Cas9 technology. So let's dive in. But before delving into the details, let me just quickly explain what CRISPR-Cas is to you. So basically, CRISPR is a naturally occurring system in bacteria that allows them to defend against viruses. However, scientists have adapted this system using the Cas9 enzyme to edit human genes with remarkable precision. So it is a revolutionary gene editing tool that has opened up new possibilities in the field of genetics. This tool is so powerful compared to the previous gene editing tools that only allowed us to add new elements to the DNA. CRISPR-Cas9 allows us to add, delete, and even replace genes. And so while it holds immense potential in treating genetic diseases, its application in creating genetically modified babies raises complex ethical questions, and this is why. In 2018, the scientific community was shocked by the announcement of the birth of twin girls in China who had their genes edited using the CRISPR-Cas9 technology. The goal of this controversial experiment was to make these girls resistant to getting the HIV infection. And in order to convert this resistance, Dr. He Jiankui modified their CCR5 gene. So CCR5 gene basically produces a receptor that allows the HIV virus to enter the cell. And so Dr. Jiankui wanted to inhibit the production of this receptor. Now, the CCR5 gene also has other important normal functions in the body, such as mediating the immune and inflammatory response. And so by interfering with this gene, Jiankui made these girls highly susceptible to West Nile virus and severe flu. So Jiankui may have protected these girls from getting HIV infection, but he made them extremely prone to getting infections that are much easier to get compared to HIV. And so this unprecedented act sparked a global debate on the ethical boundaries of genetic engineering, raising several ethical concerns. Firstly, there's the issue of informed consent. It is crucial that the individuals undergoing gene editing understand the risks, potential long-term consequences, and the uncertainties associated with the procedure. Another concern revolves around the potential unintended consequences of genetic editing. And we just discussed the prime example of this concern where the girls are going to have to worry about a relatively non-lethal infection for the rest of their lives. In fact, gene editing was incomplete in one of the baby girls, so God knows how that girl will be affected in the future. So in general, the long-term effects on the edited individuals and the future generations remain uncertain, because the complex nature of genetics makes it challenging to predict all the possible outcomes accurately. The third concern is the concept of designer babies. Currently, in vitro fertilization allows couples with infertility problems to conceive children. And during IVF, doctors can pre-select embryos with or without certain genetic characteristics, a term called pre-implantation genetic diagnosis, or PGD. And through PGD, the physician can determine whether the embryo carries the risk of cystic fibrosis, sickle cell anemia, or other genetic diseases before implanting it in the mother. The earliest case of designer babies happened in 1996 when Monique and Scott Collins decided to have a daughter through IVF to balance their family because they already had two boys before that. And at that time, this raised a lot of issues regarding the selection of traits beyond sex, like eye color, height, intelligence, athleticism, and much more. Now imagine the possibilities when IVF will be combined with a tool as powerful as CRISPR-Cas9. If we start genetically modifying embryos for desirable traits, then we risk moving towards a society where rich parents can design their children's characteristics, creating inequalities and ethical dilemmas. Another concern is that the safety and regulation of CRISPR applications are critical. And currently, there are no globally agreed upon guidelines for gene editing in humans. So establishing rigorous safety standards, transparent oversight, and responsible regulation are crucial to prevent misuse and potential harm. Furthermore, addressing the CRISPR baby controversy requires a broader ethical framework and an inclusive public discourse, because it is vital to engage the policymakers, the scientists, the ethicists, and the public to establish guidelines and ensure that the decisions about genetic editing are made collectively. Despite the ethical concerns regarding CRISPR babies, the technology holds immense potential to treat genetic diseases like cystic fibrosis, sickle cell anemia, and so on. And it has already shown promising results in lab settings and animal models. However, ethical debates should continue to shape the responsible use of CRISPR for the benefit of humanity. To conclude, the birth of CRISPR babies marks a pivotal moment in the history of genetic engineering. And while the possibilities of disease prevention and treatment are exciting, we must navigate the complex ethical landscape with caution. It is essential to strike a delicate balance between individual autonomy, scientific progress, and societal well-being. And as the technology continues to advance, ongoing discussions and ethical considerations will shape the future of genetic editing. Stay informed, participate in the conversation, and together let us navigate this transformative frontier. Please don't forget to give me feedback on this video in the comments, and stay tuned for similar informative content. I'll see you guys soon.